In this video, we will discuss about germline variant that affects tumor progression. So these germline variants is the variant that occurs within the tumor cells or close to the tumor cells, or we can say tumor uh, environment, near close to the tumor environment, in such a way that the mutation in one gene will leads to uh, the mutation in its neighboring gene, or will affect the neighboring process in a way that the future cell population will have those mutations inherently. So we will understand this thing through the several examples that are shown in this figure. So let us start with the panel 1. In this, what happens, there is a variation in the tumor cells in one gene, one allele of the gene, that is RB1. And because of the germ variant, what happens is that the another allele of the same gene will also get mutated, and from the subsequent cell cycles, or from the subsequent after this subsequent cell growth and cell division, all cells will have this kind of mutation, and the second mutation is called as SM, or somatic mutations, as this will exist in further offsprings of the cells. So as it is shown here it is mutation is only in the one allele of the gene, and after some time, or because of germline variant, the mutation is exist in the another allele of the gene also. Another kind of variant that occurs is that leads to the truncation of the protein. One example is shown here is that this gene MBD4. So mutation in one of this MBD4 gene will leads to the mutation of the other genes that are similar to this MBD4, or we can say these genes that are similar to this MBD4 will get mutated, and this mutation is mainly the result of the mutation of C with T, or we can say the C nucleotide is replaced with T in the gene, and this will lead to the enrichment, or more more genes that are mutated with this kind of variation, that leads to the protein truncating variant, or proteins are not able to translate properly. The next kind of mutation is the upregulated is related to the next kind of genetic variant is related to the upregulation of RAS target genes. So in this, what happens is this: the gene that is GRB2 will get mutated, or there is jump variant in the GRB2 gene, and because of the somatic mutation, the what happens is that this GRB2 gene will affect, or will leads to this down regulation of this final outcome, that is CIC. And how it will does that thing, it will does that thing through this pathway, that is the mutation in the GRB2, the germline variant in the GRB2 will leads to the upregulation of the RAS target gene, and this upregulated RAS target gene will prevent this CIC, or we can say the somatic mutations of this CIC will occurs because of the upregulation of this RAS target genes, and RAS target genes is also related to the tumor growth. So next kind of mutation is in the, we can say, is on the tumor suppressor gene, or the jump variant of the tumor suppressor gene will increase, or will lead to the formation of proteins in a way that protein will not be able to acquire their native structure, and this will lead to the this tumor suppressor non-functioning of this tumor suppressor gene. So this will also promote the tumor progression, as there will be no competitive proteins that are trying to reduce this tumor progression, as those proteins are become less functional with time because of the germline variant. And another kind of mutation that occurs that might lead to change the shape of the proteins, that for example is shown here, is that the cis structure of CTSW mRNA. And because of this change structure, it will lead to the genetic variation, and this genetic variation can be measured with the help of this ETQLS. This is the uh, this is the quantitative technique for measuring the genetic variant Loki. And this is also related to the tumor suppressor gene. So this CTSW is related to the tumor suppressor gene, and this mutation on this CTSW will leads to upregulation of this tumor suppressor gene. However, since there is a mutation in the structure, or it is not able to form the native structure, this will not be able to perform its function, that is to suppress gene. However, this non-native structures uh, translation rate is increased when this CTSW, when there is a cis conformation of this CTSW mRNA. And this next figure is related to the germline variant in one kind of gene might suppress the germline, might leads to variation, somatic mutation in the gene that has the similar function as that of the gene that has which has this germline variant. And one example is shown here. In this case, the mutation is GNA1, will leads to the will leads to the somatic mutation in P10 gene. And this somatic mutation in P10 gene uh, will act in a way that this mTOR pathway will become uncontrolled, as the P10 gene function is to control this mTOR pathway. uh, P10 gene is to function of P10 gene is to control this mTOR pathway by inhibiting this PIK3CA gene function. So in case of this somatic mutation, this P10 become non-functional, and this upregulation of this GNA1 will leads to the growth and proliferation of these cells, or in this case the tumor cells. Last example is affecting the splicing functioning of this mRNA. As we have discussed how important is splicing in some of our previous videos, you can check that video. So what happens in this case, in this example, is that this RBFOX1 gene, germline variant in RBFOX1 gene will lead to the mutation in other genes that are responsible, leads to the mutation in other genes, other similar genes that are responsible for proper splicing. And when these splicing genes get mutated, this will lead to differential splicing, or different combinations of the exons, and leads to different kind of generation, different uh, we can say different kind of protein generations, as protein sequence will not be same, final protein sequence will not be same as that of the original protein structure. So these are the kind of variations that might cause because of germline variant in the tumor cells, or cells that are close to the tumors, that will promote the tumor progression. So if you need to understand more detail in depth, or you need to understand more about this germline variant and its connection with tumor progression, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.